Paternity testing can be especially important when the rights and duties for the father are an issue and the child's paternity is in doubt. Tests can also determine the likelihood of someone being a biological grandparent. The comparison of physical appearance to determine paternity is probably the most primitive method. DNA paternity testing is the use of DNA profiles to determine whether an individual is the biological parent of another individual. It's the most accurate and precise technology to test the lineage of two individuals by far. The human genome contains two parts. One is 23 pairs of chromosomal DNA nuclear genome in the nucleus. One is mitochondrial DNA in the cytoplasmic mitochondria. The nuclear genome is composed of 23 chromosomes inherited from each parent. The mitochondria are all from the mother. The origin and development of the genome has built the blood ethics building of the whole human society. The parent-child relationship ultimately depends on the identification of chromosome origin. Our various phenotypes or traits are genetically determined. There are many different genetic genes between two individuals in the population, so they look different. In general, the farther the genetic relationship is, the more the genetic vibration will be. The closer the genetic relationship is, the more similar the genetic genes are. For example, the genetic similarity between parents and children is very high. So long as all or part of the genes are detected, then we can know whether the two individuals are related by making comparison. Theoretically, the more genes detected, the higher the accuracy. To detect a gene is to perform gene sequence analysis. We know that if more than 30,000 human genes are sequenced, it would take a long time and cost a lot. Therefore, the current DNA identification only selects certain loci of certain genes for analysis. Although the accuracy is less than 100%, 99.98% of them can fully meet the requirements. Microsatellite repeats are usually used for DNA paternity testing, or single nucleotide polymorphisms. Microsatellite sequence is a 1 to 4 base repeat, such as AN, CAN, CAGN, or CATGN. The number of repetition is different in population. This figure shows the duplication of two bases of CA, and the number of repetitions of three individuals, 16, 14, and 11 respectively. The repetition times of parents and children should be consistent. If multiple loci are consistent, it can be presumed that the relationship between parents and children is established. The applicant shall fill in the application form for DNA entrusted identification and the power of attorney as the basis for DNA paternity testing. After signing the agreement, blood can be taken and entered into the identification procedure. Both children and parents involved in DNA paternity testing need to extract blood. You can take blood at the site of the appraisal institution, or you can take the blood and send it for examination. For self-blood collection, only 6 to 10 drops of finger blood can be taken. After the finger blood is dried and stored, the name is recorded and sent for inspection. The staff shall number and register the blood samples after blood sampling. The blood sampling process is the same as the general outpatient department. During the DNA paternity testing, the blood samples are operated and stored by the staff, and the chances of error is almost zero. In addition, if the sample is submitted for examination, the paternity test result will be written with whether the paternity relationship between the two sample source individuals is excluded. If the evidence needs to be presented in court, the real name test is also required. After blood collection, the staff took blood samples, one of each person for verification. DNA reagents are very expensive, with a cost of 36,000 yuan. And the relevant instrument and equipment are also expensive. It costs about 3,000 yuan to do a DNA paternity test, and it is more expensive to rush the tests. The blood samples were separated by centrifuge, extract the DNA contained in the sample nucleus, and then carry out certain purification to remove the impurities in the sample. PCR was performed. The step of PCR amplification is to make a large number of copies of the fragments we need through enzymatic reaction on the PCR instrument, and amplify them to extent that they can be seen by some special instruments. And then post-PCR reaction was performed. This step is mainly the preparation stage for the detection of the sequencer. Open the double-stranded DNA, and add some internal standard for the detection, mainly to mark the length of the detected fragment. After adding the blood sample to the reagent, enter the electrophoresis instrument for gene comparison, namely the capillary sequencer detection program. Principle: because DNA is charged, the electrophoresis speed of different DNA fragments is different by capillary electrophoresis. Under the same voltage and the same electrophoresis time, the swimming distance is different. These different distances can be distinguished by the internal standard measurement added in an early stage, and it can be displayed on the computer through certain software. To facilitate the detection personnel to process and analyze the data, the inspectors analyze, summarize, and calculate the results obtained, and draw the identification, conclusion, and report to determine whether there is a parent-child relationship in the sample sent for testing.